Hello, hi everybody. Today lecture we will discuss about the Western blot techniques, one of the protein detection technique. We will see slide by slide in brief introduction and some of the methods how to detect the proteins and procedure and protocol we will be discussed today lecture. You can see the first one slide. Actually, what is the Western blot technique? The main thing is the blotting are the mainly three types like southern blotting, northern blotting and western blotting. So you can see here the blotting direction, northern blotting, southern blotting and western blotting. So here we will discuss the especially in western blot today lecture. That is the main thing is the target molecules of protein detection. The probe usually has a antigens, primary antigen and secondary antigen. You can see here the blotting are the mainly three types. For memorize, simply we can call snowdrop. You can see S means the southern blot. For D is DNA detection. Northern blot technique used for the RNA detection. Western blot technique used for the protein detection. For your memorize, you can simply keep in your brain snowdrop. Then you will move on to the next one. What is the Western blot technique? Some introduction points. Western blot is one of the often used for the research and separate and identification of protein. Nowadays you can see that day by day various techniques are coming from to detection purpose. You can see Western blot technique one of the widely used bioanalytical techniques, this one. Especially they are using in molecular biology to detect the specific protein in simple tissue homogeneity of the extract. It works on the principle gel electrophoresis. Mostly the Western blot techniques is nothing but of like a gel electrophoresis. Proteins are separated based on their size on polyacrylamide gel. You can see in these techniques mixture of proteins is separated based on the molecular weight and their by type how and they through the gel electrophoresis. These results are then transferred to the membrane producing bands for each proteins. Western blot also known as immunoblotting or protein blotting techniques used to detect the specific proteins in complex from the complex mixture of the protein. It is one of the core techniques in cell biology, molecular biology, virology and other techniques. Western blot techniques have become one of the most commonly analytical tool to detection of viral proteins. So these are the some brief introduction points for the Western blot techniques. From these we will understood Western blot techniques mostly used for the detection of specific proteins or simply you can know as the immunoblotting techniques. And you can see few more points. There are the mainly three types of blotting techniques. Earlier we discussed southern blotting, northern blotting and western blotting. While we will go to our lab, you can see that Western blot techniques where we will be detect the particular protein. So Western blot also known as the immunoblotting because here we will detect the protein by the doing of antibody interaction. That is the point is different in this technique. Suppose if you see that any other detection points like protein also detection have various Lowry method techniques and you can see the observing of the at UV spectroscopy at 280 nanometer. These are the various techniques to detect a protein, but special here is the antibody will be interact with the protein. Then after we are going to detecting the how its protein looks like, how the protein was moves into the gel membranes. This is the special technique. That's why it is also known as the immunoblotting techniques. Now you can see. Now you are the few things are remember while you are doing the Western blot techniques. Simply you can call as a precautions. Okay. So you can say during the Western blot techniques which will held in the able to remember the first you need to run the gel properly. First point is you can load the gel and you can run the properly gel. So then all the proteins are separated based on their size and there is the run of native page or the denatured gel. If you are running native page, you will not able to get the molecular weight of the protein. But if you are running the denatured gel, SDS gel, then you will get the molecular weight as well as the after you run the gel. Then you can cut the small pieces of where the protein is there for in gel or place it is the top of the membrane. There are the two types membranes, intracellular membranes and PVDF membranes. These are further we will discuss in the next slide. If you are using very big proteins, then you should be go for nitrocellular membranes. If you are using like big proteins, big KDs like 66, 70 KDs or this kind of proteins, we need to go for the nitrocellular membranes. If you are the protein is very small or medium size, then you should go for the PVDF membranes. These are the few of the points to for remembering while you are doing it to the Western blot technique. You can see next one. Actually, what is the principle of Western blot technique? How it works on techniques. So, Western blot techniques is one of the immunoblotting technique. Western blotting technique means nothing but of immunoblotting technique. While really the specific of bonding between the molecules of interact and probe to allow the detection of the molecular of interest in the mixture of many other similar molecules. The Western blot technique molecules of interest in the protein and the probe is typically an antigen or the antibody arising against the particular proteins. The SDS page gel technique is the prerequisite for Western blotting. These are the principle for the Western blotting technique, how it is works on working principle. Then you can see the flow chart, how it's the happen Western blot technique. First you can take the cellular proteins from the any different conditions, then unfold or coat it with the native charge of SDS. Then after you will generate run the gel electrophoresis separating of their according to their size. Then after you will go for the transfer of the move from to the membranes. These are the membrane transfer. Then after coat it free membranes of the generic proteins. Then after wash with primary antigen antibody binding a specific protein you are looking for. Then after wash is the secondary binding protein. Then after visualize for the visualize techniques, how your protein is, where it is protein. These are the techniques to do the Western blot. This is one of the flow chart of the Western blot technique, how what are the steps. You can see the next slide. Mainly these are the 6 steps for the Western blot techniques. First one, sample preparation. Second one is the gel electrophoresis. And sixth one, blotting, nothing but a transfer and the blocking and the antibody probing. Then after detection. These 6 points are very important while you are doing into the Western blot technique. And you can see that blotting membranes. Already we discussed in that there are the 3 types of membranes widely are using nowadays in Western blot techniques. The first one is the nitrocellular membranes and linol membranes and the PVDF polyvinylidene fluoride membranes. These are the few of the blotting membranes you can use according to our protein nature and protein size. You can see what is the procedure of Western blot technique. You can see here there is a loading matrix and then after sample loading, then according to their separate file the low molecular weight will move fast and high molecular weight is move very slow. According there you can see mobilize, immunoprobe. Immunoprobe means nothing but if you need to add the antigen primary and secondary antibodies. Then after quantitative signals will come, you record into the UV spectroscope. This is a procedure for the Western blot technique. Then what is the antibody probing roles? You can see here that once the protein sample are separated and the transfer into the membranes, the protein of interest to detect the localize using a specific antibody. Specific antibody you need to add to the membrane. The blot will be incubated into dilute solution of antibody. Usually the 4 hours at room temperature, overnight 4 degrees. This is the how to antibody probe you can add to our membrane. Then you can see the actual procedure how it looks like for the SDS pagel, then after PVDF membranes, then after primary antibody, secondary antibody with the IR dye, then after you can use the IR fluorescence detection, then after visualization of a band intensity, you can see the band intensity line 1 and line 2. This is the actual flow chart for the Western blot technique. Then after you can see the what are the steps involved in the Western blot techniques, what are the conditions to optimize. You can see that various polyacrylamide gel separation proteins, equilibrium of gel, and you can see the here the conditions and the membrane straining. You can see that this step electrophoretic transfer of membrane, blocking of free antibody sites. Then after incubation with primary antibody and washes, incubation with secondary antibody and washes and detection. These are the few of the conditions. You can see these the steps for involving the Western blot technique condition to be optimized. Then after you can see transfer of like sandwich, how it will be transfer of your protein membranes placed in the top of the gel and stuck into the filter paper placed onto the top of that. The entire stuck in place in the buffer solution which moves off the paper by capillary action, the bringing of the protein with it, filter paper and sponge. Then after you look into the how actually the membrane proteins, how to members are arranged, gel, membranes, transfer buffer and can shuttle, buffer tank, how can see members. Then after you can go with the protein transformation, how the protein will transfer from the complexion of the separation protein by polyacrylamide gel electrophoresis, the next step to the transfer of protein from the gel to solid support membranes. The solid support membranes usually made up of chemically inert substance such as nitrocellular and polyvinyl difluoride. The process of transforming protein from gel to membrane while the maintaining their relative position resolutions known as blotting. Okay. Then after you can see what are the sample loading, how to the detection of electrophoresis and the mixture of macromolecules, and you can see these. But another one precautions when you are doing to the Western blot technique, these points are very important. You should not load too much of protein in the gel of this, should be checked into the amount of protein that can be loaded based on there that you choose the sample and load that much of protein. If you load too much of protein, it will form their aggregation. If you are the loading too much of protein, then there will be oversaturation. The gel will very bad, and it also to be put on too much of substrate for their HRP enzymes. So these are the create problems. Then there will be a back spots of membrane. You should be avoid that. So these are the few of the precautions while you are doing the Western blot technique. Don't add the too much of protein. It should be check amount of protein, how it will looks, the sample will run into the your gel. You can see here the what is the few of the steps. You can see cell collection, then after cell lines, then after SDS page gel, then after electron transfer, then after antibody probing techniques you need to read out. These are the steps to involve for the Western blot technique. Then after also you can see some of the few clear techniques of point by point. You can see sample of protein preparation, then after resolving the protein sample on the SDS page gel, then after electrophoretically transfer into the fractions of protein from the gel into the membranes, then after membranes to the blocking of the membranes into the BSR milk region, then after you can see the incubate the few hours like 24 hours, then after you can go for the detection. These are the actual steps to involve for the Western blot techniques, and you can see here what are the steps. You can see the clearly how to follow by one, and you can see what is the differentiation between the Western blot technique to remember like comparison of north, south and Western blot technique. You can see molecular detection. You can see here that southern blotting, northern blot technique various. And Western blot techniques, the medical diagnostics. They are nowadays you are using various diagnostic for HIV, hepatitis B infections, and you can see the Western blot techniques used for the molecular biology and the mostly cases in protein labs in today basis because of very good techniques, and it is very sensitive to as well. So if you are want to detect any particular protein, you can detect that these Western blot techniques are one of the best technique. You can see for other techniques for to detection of protein, you can see detection of protein by absorbance at 280 nanometer, detection of protein with Lowry method, and detection of protein by the Smith-Bindix-Kulik acid method, and detection of protein by Branford Comasin Blue G250 method. These are the few other